I believe the bony sequestrum that PUSHPAK just sent in, case 569, is a very useful example to learn some things about recognizing biofilms. So this is the sample that he sent. I believe he picked this out of a bone graft site. And when you look at it, it can be very, very confusing. There's obviously a lot of cellular debris and resorption going on, as well as infection. But actually looking for biofilms can be very, very challenging in this type of a sample where there's so much going on. If you look closely, you can see, of course, white and red blood cells and a lot of connective tissue and some bone matrix in different stages of resorption. So let's try a little bit different approach here, and let's look at just some normal bone as our control and see what normal bone should look like. And Ivan sent me this case from a laser, but it had some trabeculum attached to the roots, so we got a chance to actually look at normal trabecular bone. And let's put it under the scope and see what normal bone looks like as kind of a control. And when you look at it, you can see normal kind of trabecular bone with periosteum. Looks like the periosteum is still attached, and you can see the collagen fibers of the bone and the periosteal tissue intact. Nothing terribly unusual. It looks like probably lamellar bone and maybe some bundle bone there as well. And you can see areas where probably Sharpie's fibers attached. Not a lot of cellular detail anywhere that you can detect. This looks like an area where Sharpie's fibers was attached to the periodontal ligament. And in certain areas, there are some resorptive areas, so you get a better sense of what normal bone looks like. And you can see calcified bone there, as well as resorbing bone with its collagen matrix in various stages of resorption and deposition. And when you look deeper, you can actually see where some of the decalcification is taking place, where the collagen fibers are becoming exposed. And you can see that's probably, it looks like woven bone pattern, because the collagen fibrils are pretty irregular. Before we turn to the bone sample, let's examine some dentin that we know has biofilms, so that can kind of serve as a positive control. And what we did is I took various segments of extracted teeth, cut them up into segments, and treated the canal with EDTA for 3 to 4 minutes to etch it really well, and then impregnated these samples with oral biofilm that we grew in the lab. I like these 8-well containers because they let me do each piece individually and put various tests for each piece separately and keep track of what was done. So we might have some that has a 1-day biofilm, a 2-day biofilm, a 3-day biofilm, and it's also good for its use in confocal microscopy. So there's our oral microorganisms growing in a broth in the oven, and then every so often would replenish the 8-well container with the broth material. It's the same broth that we use to actually do our testing with live dead stain. So it's kind of the same procedure we use for individual slides. And these are from our live dead stain on our broth. So then we take these dentin samples and put them under the electron microscope. But first we do some confocal imaging to make sure that a biofilm is actually present. And this is the green bacterial probe that we used here. And this is the fluorescent microscope view of that. And you can see the canal is completely filled with a biofilm. I thought I'd give a little conceptual introduction into confocal microscopy, just for those not totally familiar. When you look at these specimens under the electron microscope, yes, you get a very impressive view of what the bacteria look like. But using confocal microscopes, you get another completely different perspective because you don't have to dehydrate the specimen. And depending on the probes you use, you can detect carbohydrate, DNA, live bacteria, dead bacteria. So it's another perspective that adds a lot to biofilm science. This is wheat germ agglutinin that actually probes carbohydrate in the biofilm matrix. So now I know there's biofilm wherever that confocal lights up the wheat germ agglutinin. So it gives you another powerful tool besides electron microscopy. And for those not familiar how fluorescent probes work, the probe attaches to some protein or carbohydrate in the biofilm and then is excited with a certain frequency light called the excitation light. And in doing that, the fluorescent probe emits another kind of light that is picked up, usually a lower frequency light, as the photons strike the outer orbitals of the material. And the confocal microscope is just a series of different wavelengths lights, some dichroic mirrors that handle the separation of the frequencies, and then a camera. This is our confocal and fluorescent microscope. It's a very expensive microscope. It costs about $100,000. And then you put your specimens in it, and it excites it with the excitation frequency, and then it records the absorption frequency. Typically, these uses blue and green light. And then you get a readout from the camera of on the absorption of the light. And the probe basically can indicate a great many things depending on the probe. This is the interface on the microscope. So you do this in layers. And then the probes themselves are expensive, about $500, $500 or $600 a package. But there's all kinds of different probes that you can use to tag whatever you're particularly interested in. We use green bacterial stain. We use live dead stain. That's live dead stain. And then we use a carbohydrate stain, a lectin, to stain carbohydrate in the biofilm. And this is done for looking at the culture bacteria. These are from our culture bacteria. But we're going to use this in conjunction with correlative electron microscopy. So when you look at a specimen, now I've used the green fluorescent probe, and now I know that that canal is filled with bacteria. These are confocal microscope shots of what the canal actually looks like. Anything that's green is bacteria. So I know the canal now is filled with bacteria. And then when you go in and try to correlate, well, where is that bacteria? It'll give you a sense of what the bacterial signatures actually look like, because you know that there are bacteria there. And when you look at it, you start to pick up and recognize these signatures eventually. And here you can see it. You have to look carefully. And in the Google group, when you look at the images, I've given you many, many different powers of images that you can use your scroll wheel, and you can zoom in like this and see if you can start to identify a bacterial biofilm signature. Here you can see a very good signature on the dentin. You can see the EPS has a certain signature to it. It looks like molasses, melted molasses. And once you start to recognize this, it'll be easier for you in looking for whether a material has a biofilm on it or not. So I ask you to kind of look at these images carefully. Here you can see some planktonic bacteria actually on the surface of the dentin. But most of the bacteria is in this EPS, and what you're looking for in your signature is some kind of form that indicates there are bacteria within the EPS. And here you can see, you can make out the bacterial morphotypes embedded in EPS. And without that signature, it's very difficult to actually tell that there's any bacteria in this canal at all. So these are various different views of those samples that I made from cut teeth that are filled with biofilm, but it's not immediately obvious that it's biofilm. You have to look for it. You have to search for it. You can see what the EPS looks like. Here's an area that just looks like kind of like a smear layer, but if you zoom in and look very carefully, you can see a classic biofilm signature of the bacteria embedded in this EPS. Again, it's unmistakable bacteria in a biofilm. So when you look at these, start to refine your pattern recognition, and after a while, you'll see the signature almost immediately. Even at low power, you can kind of start to identify likely spots where a biofilm is present. These are not like pictures in the JOE where everything is obvious. You have to do a little work and actually look very, very carefully. They're not planktonic bacteria. They're embedded in this EPS, and you have to be careful as you look at these. I've tried to make some examples where maybe it's a little more difficult to look at this. When you look at this, you're looking in a dentinal tubule. It's not immediately obvious that there's biofilm there. You see the calcospheroids from the decalcified human dentin. But if you look carefully, you can see that melted molasses look to it with bacterial morphotypes embedded within that matrix. And now your level of suspicion is really raised when you see that. So here it's difficult to tell, is this just decalcifying dentin or is it a biofilm signature? And when you spend time and actually look around, you will eventually find some bacterial morphotypes in this EPS, and that can help you really identify it. Here you can see the signature of a biofilm. It's very, very clear. So let's go back to the original bone sample now and see if it doesn't help you a little bit. And obviously you have a lot of red blood cells and white blood cells, and then a lot of tissue and fibrous connective tissue. And your job is to see, well, is there any biofilm here? And obviously we know that there is because this is infected bone, but actually localizing it and certifying it is a little difficult. So there you can see some red and white blood cells, but if you look very, very carefully and zoom into this area, you can see the unmistakable picture of bacterial biofilm in EPS virtually everywhere on this sample. Everywhere you look, you'll see this EPS and bacterial morphotypes embedded within the EPS. So I hope this helps you as you go through these pictures in the Google group. Start to train your eye a little bit. Resorptive areas are always good to look at. Here you can see a lot of planktonic bacteria present sitting on the decalcifying dentinal matrix, probably playing a role in the decalcifying of that matrix. Here's another fragment from PUSHPAK'S case, and when you go into it and really look at it carefully, you have to look carefully. They're just not sitting on the surface as planktonic bacteria. But when you zoom in these areas, there's the bone matrix that's pretty well decalcified. You don't really see any calcospheroids. And then when you look carefully, you can see all kinds of bacteria, basically everywhere on the specimen, inside the specimen, in all locations in this EPS matrix. So I hope that was helpful to you in starting to train your eye to see the pattern recognition.